It's my great pleasure to introduce our speaker for today. Pieter De Beule is a staff researcher at the International Iberian Nanotechnology Laboratory, or the INL, in Braga, Portugal. His research team there focuses on developing new optics-inspired technologies for the nanosciences. Pieter holds a master's degree in engineering physics from Ghent University in Belgium, as well as a PhD degree in physics from Imperial College London. He's done research in the UK at the Max Planck Institute in Göttingen, where he's developed new microscopy approaches for fluorescence lifetime imaging, as well as confocal microscopy. Pieter has been at the INL since 2011, where he's been working on implementing new imaging systems, combining atomic force microscopy and optical microscopy. And this is also what the topic of his talk today is, where he'll be telling us about simultaneous and co-localized imaging of fluorescence and atomic force microscopy. Thank you, Pieter, for joining us. Really looking forward to your presentation. And please start your screen sharing.
Once again, welcome all to this presentation, and I would like to thank Johanna for extending me the invitation for giving a talk today to put our work a little bit within the context of how to. So I will be talking to you today about correlated microscopy, and in specific correlated microscopy for combining atomic force microscopy for biological purposes with optical microscopy as a whole, and in particular at the end of the talk I will focus on some of the developments in combining atomic force microscopy with super resolution fluorescence microscopy. And so I will give a bit of an overview today. I will talk a bit about the challenges. So I will assume that well, I will try to explain what are the challenges if you would like to make a setup like this in your lab, or even you would like to explore certain things, that there are limitations to certain aspects, and you should be, well, it was nice to be aware about it before engaging in this type of work. Okay, so just as a small introduction, so I am based, as Johanna said, at the International Iberian Nanotechnology Laboratory. So we are based in the north of Portugal, in Braga, actually close to the Spanish border. And so, yeah, we have been operating since 2011, and we also have a few facilities, and one of them is focused on bioimaging. So actually, I had to think a little bit, because earlier this year I gave a webinar that you can also find online and you can get access to. And so I realized that this webinar was kind of, it was geared towards the atomic force microscopy community. And I realized after Johanna invited me that today, I could change the webinar a little bit and focus it more towards people that are working in the field of fluorescence imaging and also maybe electron microscopy, but so which I believe is a bigger base of the people active in the Euro-BioImaging community. But so, okay, let's get started today. But what could be the motivation of using two microscopy techniques at the same time? So the idea is general, that you would like to gain more information about your biological sample, and you would like to image preferably at the same point in time, and you want to extract more information about what goes on into your cell or your bacterium or other biological samples. So I mean, here you see a little example of how a typical image of a fluorescence image overlaid with with an AFM image typically looks like. So typically, the AFM image is a little bit smaller than the field of view offered by the normal high aperture numerical aperture fluorescence images. And so the thing is, well, what you can wonder here, so here you see the topography of the cell, while the purple reflects, I think, the actin filaments within the cell. So you can ask yourself, okay, what is the use of that, or let's say, how can we extract useful information then of combining these two techniques? So I'm today going to explain a little bit more about this, and I wanted to actually position also a little bit the atomic force microscopy combination with fluorescence and also label-free imaging within the context of all the possible combinations one can think of. And so over the last 2 decades, I believe there has been a lot of progress and reports in the literature combining the microscopy technique. So either scanning probe microscopy with optical microscopy, or then you also have electron microscopy with optical microscopy, which is CLEM. And so today we're going to focus then, of course, on atomic force microscopy combined with label-free and fluorescence microscopy. So I got in contact first with Euro-BioImaging myself when I attended an event of the Comulus event, which I believe now it was a cost action then that later on continued, which I was very happy to hear about. And the thing is, I put a little question mark here because, okay, people have published a lot of reports, and in the end maybe you could think about combining all of these techniques at the same time, but there are of course restrictions to that, and that hasn't been actually demonstrated yet. So just setting the scene a little bit, so we are talking today about how to integrate an inverted optical microscope with bio-AFM system, okay? So typically you have to use an inverted optical microscope, of course, in order to fit the bio-AFM. So this typically, this stage replaces the motorized stage of the fluorescence microscope, and you have to find a way how to fit this system underneath. So one thing that, well, you can see that you have to have enough space for that. And also the stages have to match. So when you acquire these systems, you have to watch out a little bit what you acquire so that the holes fit properly. Okay, so but one comment regarding the previous slide regarding is that you actually do need a long distance condenser for the imaging of when you are inserting your atomic force microscope, because that's typically the height of the atomic force microscope is around 7 to 8 centimeters. So the most of the high performance fluorescence microscopes, they have a condenser with a working distance of somewhere between 2 and 3 centimeters. So you do need to replace that unit in order to guarantee proper color illumination. So this is what I just wanted to highlight here with this slide. And so now I'm going to, because I'm under a little bit of the assumption that the audience today might not be so familiar with the latest advances in bio-AFM. So I wanted in the next few slides explain a little bit what bio-AFM has to offer for biological systems. So most of you might know that you use an atomic force microscope for making topography images in the nano scale regime of any object you can find out. But so for biological purposes, there are also several other features that extend to a greater the capacity of the bio-AFM. And so one of them is basically something known as force distance imaging. So force distance imaging is a procedure whereby you approach with your AFM cantilever a surface, as you can see here on the bottom right. So I'm gonna see if I can make a pointer. Okay, so here on the bottom right, so basically while you approach and you have here the blue curve, your AFM cantilever, you start indenting your sample, and then when you record the force experienced by the AFM cantilever while you retract, you typically have you can monitor a certain adhesion to the surface. Okay, so and also if you fit the compression part of the curve, you can extract elasticity properties of your sample. So this gives access on a nanometric scale to the nanomechanical properties of your sample. So as you can see here, then you can create an image not only of the topography but also of the elasticity of your sample in an image view, and also you can monitor the adhesion to your sample. And so depending on the substrate that you have underneath your cantilever, these things will change. And so then of course also another thing that you can consider if you're interested in bio-AFM is that you can convert your AFM cantilever that monitors your topography or your distance force distance curves, and you can attach almost anything you like to these cantilevers. So the reference that I placed here gives a very good overview of how you can modify your AFM cantilever and convert it into some sort of biosensor. So you can attach proteins, viruses. I think almost anything you can think about, you can almost attach it to a cantilever and use it to do force probes with this. So, and then another thing that maybe is worthwhile mentioning, because this is so a normal atomic force microscopy, for if you want to image a biological cell. So if you want to make a high quality image, the acquisition speed is of a normal atomic force microscope relatively small, meaning that a high quality image can take you 10 minutes to acquire. So around 20 years ago, the laboratory of Toshio Ando in Japan made several steps forward in trying to improve the imaging speed of the atomic force microscope. And so this was done in three ways. One was that the electronic circuits was heavily modified in an analog way. The scanning stage was also changed, and also because you want to have a higher scanning speed, so you need to scan your cantilever much faster. So you have a highly reduced size of the cantilever. And so with these advances, people have been able to monitor the movement of, for example, myosin 5 on actin filaments, as you can see here on this reference of this paper in Nature. So so you can see here what you should maybe remember is of this image is that the difference in time scale between these images, you can see here 147 milliseconds and 293. So you go from an imaging acquisition speed of between 10 minutes to down to sub-seconds, and this can be several hertz, depending, of course, on the size of the image that you're trying to scan. So this advance of high-speed atomic force microscope has been for a long time, I believe. Maybe a little bit wrong in this, but it has been for a long time limited to several laboratories that were really experts in this. But in the recent years, there have also been a catch-up of the more commercial manufacturers of atomic force microscope for biology purposes to also implement some of these advances. And then so lately, I also wanted to explain to you a little bit about what is the kind of resolution you can get from a bio-AFM. So one advance that was recently published by the laboratory of Simon Turing in Cornell University. So it brought the typical resolution limit of a bio-AFM is on the order of maybe 5 to 10 nanometers or a little bit less. It depends on how sharp your AFM tip is and what the radius is. Okay, but a few years ago, so the laboratory of Simon Turing had the idea, so they converted the concept of what you probably know as photoactivated light microscopy of stochastical optical reconstruction microscopy, storm imaging, or Palmer storm imaging. They converted that concept to the field of bio-AFM. So instead of switching on and off the fluorophore that you have, as you can see here on the left-hand side, people were looking at stochastic fluctuations in the height that occur if you image a sample multiple times. Okay, and so as they are so they in this way they managed to bring the resolution of the bio-AFM in the sub-nanometer regime. So people managed to actually, so what you see here on the bottom left is an extract from this paper, and you can see that how they for aquaporin, which is a molecule that transports water across the E. coli membrane. So you can see that they managed to get a match between molecular dynamic simulations, I believe, and so they managed to locate the amino acids inside this protein, and they found a match with the topography image that they extracted with what is localization, what they call localization atomic force microscopy. So, well, so this is a relatively recent advance. So in this case, one difficulty, of course, is that if it relies on imaging your sample multiple times, and that's in the atomic force world, it's not so easy. So even though this concept can be applied to high-speed atomic force microscope or normal speed atomic force microscopes, you remain with this challenge. And actually, of course, when you scan faster, you distort your sample less, then it's more easy, of course, to extract this information. So this introduction was a little bit, so I hope I explain you a little bit more about the world of bio-AFM and what are the recent advances and what you could expect from an atomic force microscope. And I also wanted to stress that it's, of course, it's not only limited to monitoring topography. Okay. So, okay, now let's focus now on let's say what we're gonna explain about. So about how to integrate atomic force microscopy with optical microscopy. So basically, one thing that one reflection, and given that this is a pub session, so one reflection that I have sometimes when I see reports or papers show the field of typically one research laboratory is expert in implementing one type of this combined microscopy, and what in the broader community maybe is missing is a very fast way of benchmarking these results, because you have to really dig deep into the papers to compare the different papers and the different technologies, and it's not always so easy. And that's maybe because in the broader community there is no easy parameter that allows you to compare these results in an easy way. So one thing that we should consider here is that to what extent do the frame rates of the two imaging modes overlap? So typically, well, as I mentioned already before, the atomic force microscope is relatively slow compared to the fluorescence microscopy or even optical microscopy. And then so the lengths of monitoring, they can be quite different between the two technologies. So, and then also in space, you have to really observe what is the spatial overlap of the volumetric overlap of the areas you're monitoring. And so that there I maybe want to refer to the image I showed you first. So you see a topography image, and you have underneath a picture of the actin filament, and you can wonder, okay, what is the point of having a topography image and underneath the actin filament? So sometimes there is a bit of an overlap between the two imaging systems, or sometimes there is simply none. And then one thing also we have to bear in mind is that, well, an atomic force microscope is a system that is highly linear when it comes to movements in X and Y. And so that, as I'm sure you know, in terms of microscopy, it is not always so perfect as you would like it to imagine. So many optical objects, they have optical microscope objectives, they have a certain level of geometrical aberrations, and depending on the choice of the objective, they are quite different. So and then lately, and we will explain about that later on, so you also have to bear in mind that if you operate one device, so let's say your imaging, your optical imaging, for example, can induce distortions in your atomic force microscope, and vice versa even. So because there is even the movement of a camera fan in a inverted microscope, it can distort the movement of the atomic force microscope cantilever. And then one thing that I will focus on a little bit later is also there is a possibility of thermal heating that occurs in the vicinity of the atomic force cantilever. Okay, so now I give a little bit of a brief overview of some achievements from the past and things that are maybe kind of relatively straightforward. So you can combine relatively easy. So you can do reflected dark field microscopy or reflected DIC microscopy, and you can overlay it with atomic force microscope. This has been demonstrated in the early 2000s. And then so I also wanted to briefly mention, so because I know that this is a very, let's say, a hot topic, because there's a lot of interest in the imaging community, is in quantitative phase imaging. So this type of technology, of course, it is not really possible to integrate with a atomic force microscope in an easy way, because you will always have the quantitative phase imaging, or at least the ones that I'm familiar with, they are relying on a transmission geometry of the illumination and the detection, as you can see here, from this review paper by Popescu, Park, and De Beule. So yeah, so this is not really a viable route to implement with in an easy way with atomic force microscope, and to my knowledge has also not been demonstrated. But one interesting route for the future that I thought of, what people could explore hopefully, is the combination of something like rotating coherent scattering microscopy. So this is a concept brought forward by the group of Alexander Rohrbach in Freiburg. So in this case, what you can see here, when you illuminate basically with a laser that is rotated in backfocal plane at a very high speed, you obtain different scattering images of your sample. And so the rotation happens incredibly fast, and it allows the reconstruction almost at 100 hertz of very high resolution image of your sample. So this is of course interesting to integrate with atomic force microscope, because you have very limited amount of light that is being transmitted to your atomic force microscope cantilever. So this is maybe something interesting to consider for the future. So now let's focus a little bit on fluorescence microscopy. So basically here you see all the improvements in imaging that you have, of course, with going from a wide field fluorescence microscope to a confocal type confocal microscope. And so here on the left, I just try to visually focus your mind a little bit on where where the bulk of your excitation light is, and where your imaging is retrieved from. So if you have a confocal-like microscope, you can, of course, position your imaging plane at different distances with respect to your cantilever. So your cantilever doesn't have a lot of room. It will always be on the top leaflet of a membrane leaflet of a cell, while you can vary quite easily the imaging plane of the thing, and that of course will determine your volumetric overlap, which is, I mean, this is relatively straightforward, but I wanted just to focus your mind a little bit on that. So one thing is that one thing to bear in mind, and I know that the integration of confocal microscopy with BioIFM has been relatively popular, but I just wanted to point out that typically when this integration is done, and you are operating both the AFM cantilever and the confocal microscope spot at the same time, you have to bear in mind that typically when the confocal laser scanning spot hits the cantilever, the experimentalists, they remove the data point, and that's because the light contained within the confocal laser scanning microscope, it is relatively powerful, especially since it is focused, and it distorts the movement of the atomic force microscope cantilever. So the question also that I will also talk about later is that how can you extract information in the fluorescence information at the same kind of position of your atomic force microscope cantilever? Because that's, of course, I mean, ultimately one can think of that is what makes sense when you consider combinations of the technique, because you want to monitor in the same neighborhood or the same vicinity, because otherwise one might argue, okay, we can just operate these two imaging modes on two different systems. So what would be the need of integrating them in a single one? So, I mean, in this graph here also, I wanted to illustrate you that the illumination of the FM cantilever is, of course, in time, it is not constant. And so basically so what we noticed is that, so we have, in our lab, we have a confocal-like illumination system that, so basically, in our case, the illumination is around 175 microwatts at 560 nanometers. So, and we found out that the continuous wave illumination of this cantilever induces a bending of 9.1 nanometers. And so then basically, if you consider how this cantilever is built up, and if you assume a two-dimensional simplification of the cantilever, so in this way, we were using here a triangular cantilever, but if one reduces this to a two-dimensional model, there has been a research publication dating back to, well, it is 1925. It's not a typo. We are going to celebrate soon the 100th year of this paper. But so this paper was making a link between the mechanical deformations and the temperature increase that is induced inside the cantilever. So what happens here is you illuminate the cantilever deflects because it is a bimorph. So basically, the cantilever consists of two metals, so in this case, gold and silicon nitride, and they because of the heating, they expand in different ways, and this induces a permanent bending of the cantilever. So this paper made a link between the deflection that you observe and the temperature. So basically, if we plug in these numbers, we were observing a temperature rise of 0.33 degrees Celsius. So this is not negligible, and I'll explain also later. So basically, but there is one easy way that you can do so in atomic force microscopy. Well, so the cantilever that you see here on the left, this is how to say a relatively cheap all-purpose cantilever, but you can of course reduce or completely annihilate this phenomenon by using what is known a Euro-BioImaging cantilever. So there, the gold coating is not continuous across the cantilever, but you only have a gold coating on the very edge of the thing. So basically, we as this was a two-dimensional simplification of the system, we actually did some simulations with Lumerical, which is a software that allows you to completely model the cantilever and the illumination. So basically, so this is what we found out, so if you illuminate your cantilever with 0.3 milliwatts at 560, basically you have heating that occurs in the range of fractions of a degree. But if you illuminate with 1 milliwatt at 405 in a confocal mode, you can see here that your illumination, your temperature increase that you induce is considerably higher. So then you go up to 10 degrees Celsius or Kelvin, let's say. And so, yeah, here on the right-hand side, you see the type of power fraction that is absorbed by the gold layer of the cantilever. So I also want to make one small remark here. So it is possible to perform bio-AFM with a cantilever that doesn't have the gold layer. But these cantilevers do not perform that well. So they have to a certain extent reduce performance, because it is so you still need to reflect within an aqueous environment. You need to reflect your AFM cantilever, right, which is always in the near infrared. So this red laser light here is typically around 800 nanometers. So yeah, the gold coating, it is quite difficult to replace this with something else. But if you replace it, usually it's also another metal-like structure that also is affected by heating. But gold is the most used system. So one thing that we noticed in our simulations, and this might come a little bit as a surprise. So basically, when so we noted that the heating. So if we assume that the dish is the point of thermal equilibrium, we noted that the temperature increase induced by the laser is heavily influenced by the distance of the cantilever with respect to the bottom of your dish. So as you can see here, as you increase the distance of your cantilever with respect to the bottom of your dish, the greater your heating of your cantilever will be. So that means that if your cantilever is very close to your dish, you will not experience so much heating, and that would be as if you do atomic force microscope on proteins that you attach to the surface or something like this. When you do this kind of molecular biology, then there is not such a big impact for the heating. But if you try to monitor the top of a living cell, for example, and then you maybe have several nanometers, several micrometers, then the heating, it is greatly affected. So what you see here actually on this graph here, it is, so the parameter D is the distance of the AFM cantilever with respect to the bottom of the dish. And what you see here also is that the heating across the cantilever is not completely constant, but well, you can see the curves are maybe relatively flat, but not completely. Okay, so then basically, if we summarize this and we look for the effect of the distance of the cantilevers, you have here in these graphs. So on the left, so the left two graphs, you have the P and PTR cantilever. This is this triangular cantilever you saw before, and then on the right hand side you have the Euro-BioImaging. But so what actually is the take-home message of this cantilever is that, okay, of course, if you are doing confocal microscopy slightly towards the red, the impact on the heating of your cantilever will be reduced quite heavily. But if you use wavelengths of 488 or 405, you actually observe a relatively strong heating of the cantilever. And so, but why is this relevant? Is because if you're trying to image living biological specimens and you're trying to monitor this in a Petri dish heater, and you're trying to establish 37 degrees, and you're hoping that what you're imaging is going to be completely within physiological conditions. So maybe if you, and I'm not saying that this always happens, but you have to be careful that you don't induce an unwanted heating in your AFM cantilever when you're using at the same time fluorescence microscopy. So in our case, one implementation that we did is that we used a little bit less known technique of differential spinning disk imaging, which is basically an alternative way of obtaining optical sectioning in fluorescence, not using the confocal microscope technique. So in this case, so this stems back to a paper from the group of Tony Wilson and Mark Neal, and together with Rimas Juskaitis, sorry for if I don't pronounce the name correctly. So basically, they introduce a random illumination with a spinning disk, and so then the spinning disk that you see in this image is actually the same physical disk inside the microscope. So but basically what you should bear in mind is that you collect on a single camera, you collect a so-called conjugate and a non-conjugate image. And if you do imaging overlap, you overlap both images, and then you do a scaled subtraction usually, then you can extract the optical sectioning information in your fluorescence mode. So this is an alternative way of obtaining optically sectioned images in fluorescence microscopy. But we found out that specifically for the problem that I was talking to you before about the non-continuous illumination of your AFM cantilever, but these two graphs are a little bit simplified, of course. But so if you illuminate the cantilever, AFM cantilever in a confocal like microscope, the illumination is non-constant and it induces distortions. But if you actually use the DSD principle, you have actually on the time scales of the movement of the cantilever, you have a kind of a constant illumination of your cantilever. And this constant illumination is what induces a constant bending, and it does allow for the movement of the atomic force microscope in a proper way. So this is the kind of system integration that we have here in our institute. So yeah, there are, of course, so on the left hand side, you see the picture and the diagram. So you have to bear in mind several things that you have to eliminate. So because also the spinning disk induces movements, and you have to mitigate that the movements of the mechanical noise induced by this gets transferred to your AFM cantilever. So in this case, you could see we were trying to image a piece of mica, and it was probably a bit of a dirty piece of mica because you had a bit of height fluctuations here. So, but when we switched on the DSD disk movement on or off, we were observing a kind of an induced error, let's say, of around 10 nanometers peak to peak, and that's of course very problematic if you want to do atomic force microscopy. And so this you can see here on the right hand side, this noise that was introduced in the AFM had around 100 hertz, which is what we read to be the actual movement speed of the disk inside this DSD. So well, doing some engineering, then we were able to eliminate these noises, but still this is not trivial, let's say. I mean, you have to spend a bit of time to optimize this. So this is just to point out that if you expect to purchase or acquire these systems, and just plugging them together, it's not so easy. And then of course one thing that I have not yet seen as of today, which is the integration of atomic force microscopy with basically the so-called, this is a commercial name, but basically it is whereby you shift a sinusoidal grid across your sample plane, and you move this 3 times, and then you also can obtain an optically sectioned image in a fluorescence mode. So this has, of course, resemblance with structured illumination microscopy, which I will get back to later. But typically, in this case, the spacing of the sinusoidal grating is much larger than in the case of structured illumination microscopy. But this approach probably could be interesting to implement with atomic force microscopy as well. And then, so maybe one thing that I wanted to just also briefly mention is that because I thought in this community there are quite a few people interested in light sheet microscopy, is that there was also a report by the group of Richard Superfine of integrating atomic force microscopy together with light sheet volumetric imaging. And so they presented a very interesting approach whereby they lowered a prism next to the sample that they were imaging in combination with the atomic force microscope. So if you have any interest in that, I would suggest you check out this manuscript here. I have to say that when I was analyzing the imaging itself, it is not so straightforward. So as you can imagine, if you want to replicate this in your lab, I think that the prism is going to pose you some challenges, and how to position the prism in the correct way. I think that might be a bit involved, but feasible, of course. And so just summarizing this, so if you can combine AFM with all these fluorescence microscopy techniques, and then but of course some of them have some advantages with respect to the others. But for simultaneous image formation, the differential spinning disk, which just cannot be confused maybe with let's say the other the NIPCO disk configuration, because sometimes the imaging formation in the NIPCO disk for multiplexed confocal imaging is very different from what is known to be the differential spinning disk. So then the apotome system, that combination hasn't been explored yet, and so of course the light sheet microscopy that might also offer some interesting possibilities for some of you. So now just going back to the combination of atomic force microscopy with super resolution microscopy. So a few years ago, we wrote an extensive review on the topic, and you have the reference there, and we tried to summarize all the advances that have been made in the field. So basically, combining far field super resolution microscopy has been started more or less around 2010. But before that, people were already exploring a more near-field fluorescence super resolution approaches. And so as you can imagine, integrating atomic force microscopy with total internal reflectance fluorescence microscopy is relatively, in terms of illumination, it is relatively straightforward, because on the side of where your AFM cantilever is located, you obviously only have the evanescent field. And the evanescent field is not powerful enough to distort your atomic force microscope motion. So in that sense, that's a very interesting approach to combine. But that of course is limited to samples that are suitable or amenable to the TURF imaging mode. So you cannot be more than 100 nanometers away from your substrate. So very interesting, but some restrictions, of course, related to the sample that you might want to explore. And so basically, one of the challenges, for example, so now I will talk a little bit about the integration of atomic force microscopy with the three kind of maybe traditional super resolution techniques, which is palm storm, STED microscopy, and then also structured illumination microscopy. So the first thing to know is that if you want to integrate palm or storm imaging and with atomic force microscopy, of course, conceptually it's very intriguing, because you bring the resolution of your atomic force microscope in the same order of magnitude as your fluorescence imaging. So in terms of matching the resolution of the two imaging systems is very interesting. But the system integrations are complicated by the fact that you typically need a relatively high power laser power for the imaging of the single molecules, and that can lead to cantilever disruption or even permanent damage to your cantilever, of course, depending on the amount of light you pump into your cantilever. And an additional problem in the case of combining storm imaging, so is that the actual buffer used for storm imaging, which is the oxygen scavenger, it is problematic for the physical integrity of your AFM cantilever. And so actually a very interesting alternative approach has been devised by the group of Susan Cox in collaboration with Lisa Hirvonen, who is now working in Australia. So they found an alternative way to the oxygen scavenger typically used in the storm approach. So yeah, so they applied this to what is known to be THP1 cells. I mean, as I'm not a biologist myself, this is I'm always a bit puzzled sometimes by these acronyms. But yeah, so they monitored two-color storm imaging, and they managed to combine the data with atomic force microscope in a simultaneous data acquisition. So an alternative approach, of course, is offered by STED, stimulated emission depletion microscopy, and so this has been pioneered by the laboratory of Alberto Diaspro in Genoa. So back in 2012 they demonstrated the integration of STED microscopy with atomic force microscopy. But one issue, and the problem here again relates to the illumination of the of your AFM cantilever, is that this has also been demonstrated by the group of Clemens Kaminski in Cambridge. So one issue, so even though some form of simultaneous operation has been demonstrated, it remains a problematic thing that the depletion laser beam is, as you know, is in order to be effective, as you know, is very powerful. And so this type of depletion beam you have to be very cautious with, because it can lead to the complete disintegration of your AFM cantilever. It can be completely annihilated. So if you want to combine the two imaging modes, presumably you have to make sure that the two the light beam and the FM probe itself, they don't come in collision. And then also one interesting prospect I have been debating a little bit about it is that okay, so recently there has been a lot of advances in mitigating this strong laser beam, and people have been exploring min-flux as an alternative. So yeah, here, of course, in this case, you still have your donut beam, and you're trying to look for the minimum in the fluorescence. And so this also has been demonstrated to lead to very high spatial resolutions, and also even temporal resolutions, as demonstrated in some papers in Science from a few weeks ago, or let's say earlier this year maybe. And so yeah, but interestingly also this approach was demonstrated on a similar type of kinesin motors that were moving on actin, similar to the high-speed AFM images that I've shown you before. So well, this is it's an interesting consideration. So so let me move on in the interest of time and close here. So then we thought, okay, but what about integrating atomic force microscopy with structured illumination microscopy? So as you know, you cannot get the same type of resolution enhancement with this technique as with the other two techniques. But it has a kind of an advantage, of course, that the illumination density is typically lower for live imaging. And so there was a very nice talk given by Eric Betzig a few years ago in Vancouver, who pointed that out in a relatively, consistent way. And he pointed out that, yeah, sim microscopy only offers you, in terms of point spread function, reducing by a factor of 8, so back a factor of 2 in each dimension. But it does allow for a much lower light illumination that then allows for longer time imaging of living samples. So we thought, okay, what about doing that type of integration with atomic force microscopy? And it turned out that, so in this case, we established a demonstration of integrating this super resolution structured illumination microscopy. And so we could not see, while we were doing the atomic force microscopy, we could almost not see any difference in the performance of the atomic force microscope with the sim illumination on or off. So this was illustrated by this image here. So okay, we saw a very slight increase in noise maybe, but it's on the order that it was not really relevant for the type of structures we were observing. So just to conclude maybe, so this is what I was thinking could be a very interesting approach of, let's say, that people could develop further for imaging interesting processes in biology. So it's like one is capable, of course, as I mentioned before, to modify your AFM cantilever and to convert it into a biosensor, and you can attach any ligand to your AFM cantilever. That could, in theory, activate membrane receptors, and that they could activate a downstream signaling pathway that then could be monitored by your fluorescence microscope. And so if you would develop an experiment like this, I think it could reveal maybe some outstanding questions on kind of signaling processes in cells, whereby sometimes some dynamic information is restricted or even not available maybe. So of course, further pathways to improve on the demonstration that we did a few years ago is to move to high-speed atomic force microscope to bring the imaging times of the atomic force microscope in the same order of magnitude as the SIM imaging. And you could also go to video rate SIM imaging, which is the group of Thomas Hooser demonstrated also a couple of years ago. So just to finish off here briefly, because I know we are running out of time. So what we are currently working on is that we are going to implement a new way of doing structured illumination microscopy, or in collaboration with Thomas Hooser from Bielefeld, to use an all fiber-based SIM system for the integration with atomic force microscopy. And so actually, I just wanted to make a small announcement here. So we will, in brief, we will open a few positions related to this work of combining atomic force microscopy with super resolution structured illumination microscopy in the field of so in the context of a European doctoral network. So okay, I think with that, so I will just briefly conclude that. So the perfect the perfect imaging system for your sample, it doesn't really exist. I mean, you have to consider a lot of things, but so of course, I think I will conclude here with this slide. And okay, thank you all for your attention.